started.
Hi, everyone. Thank you, Ella, for the introduction. I'd like to thank the former committee as well, Sam McQueen and Kristen Lee, and also Gabriela Dodd and Condelo for the invitation and the opportunity to present here. I'm super happy to present for you as I was a CGIL, and I think you should be seeing my screen by now.
We see your screen, but not the,
Okay.
not in presentation yet. And yeah, all good.
Okay. As Gabriela presented me, I'm Larissa Graciano Braga. I am a PhD candidate in animal science with Dr. Denise Munari at São Paulo State University in Brazil, and I was a visiting student at CGIL under supervision of Dr. Flávio Schenkel, and I'm super happy to be here presented for you. These pictures was this year in Alberta, and I really miss you all, everyone. I learned a lot from you. And today I'm going to talk about a paper from my master thesis that is entitled Identification of Copy Number Variations in the Genome of Dairy Gir Cattle. So the Dairy Gir is a Bos indicus. These populations come from the Gir breed this originated in India, and there was some importation of Gir animals in Brazil in the 19th and 20th centuries with fewer than 700 individuals. And at the beginning in Brazil, those animals were raised as dual purpose, but then some breeders started to focus the selection towards improving milking threats. Then the National Dairy Gir Breeding Program was created in the 80s with a focus to optimize milk production in the Gir, then establishing the Dairy Gir population. The Dairy Gir cattle is well adapted to the tropical conditions. It has tolerance to parasites and also to heat stress. And that's why it is an important genetic resource in the tropics, being raised in Brazil and other South American countries. Due to its role in the tropical milking industry, it is important to characterize and to understand better the genetic architecture of those animals. And a way to start studying the genetic architecture of those animals are to study the structural variation in the DNA, such as copy number variations. Copy number variation stands for CNV, and it involves deletions and duplications, ranging from 50 base pairs to several megabases, with variable copy number compared with a reference genome. There is a debate in the literature whether it's from 50 base pairs to 1,000 base pairs. And when we observe CNVs happening in the same region in multiple individuals, we then can establish a CNVR region. So in order to do that, we identify individual CNVs, and then we can overlap them by at least one base pair. To exemplify what is a copy number variation, here you have a figure. Starting with the normal state would be a copy number S2. I have both alleles in each homologous chromosomes. In this state, I have no copy number variation, and this would be the reference. I can also have a deletion in only one homologous chromosome, or I can have a deletion in both chromosomes. I can have also a copy number S2, but with a CNV where I have a deletion in one, and in the other chromosome I have a duplication, and I also can have a copy number S3 mixing deletion in one homologous chromosome and duplication in the other one. And with the current methods, we can detect up to 4 copy numbers, and we can have mixtures of duplication, deletions in both homologous chromosomes. So we see that copy number variants, they can be large in size. They are up to several megabase. So it can change a lot of a big stretch of the DNA. CNVs can alter phenotypes and alter proteins by doing gene dosage. As an example, a duplication can increase or a deletion can decrease a copy number of a gene. It can also modulate the gene expression. A deletion could disrupt a start or stop codon. A duplication can alter a promoter region. It can also change the modulation of gene transcriptional regulators by deleting or duplicating the binding sites of transcriptor factors or even gene silencers like microRNAs. As an example of how a CNV can affect a phenotype, I brought here an example in pigs with coat colors. Here I have this picture, this figure. As legend, we have here DBB, which will stand for a breakpoint of a duplication. Here I have in red a key gene, and here the star will symbolize a mutation. So we have the wild type, that is the key gene without any mutation or duplication. This is for the wild board. I have no duplication in the key gene or mutation. And when there is a mutation upstream and downstream KIT, we'll have the belt phenotype that we can see in the Hampshire breed. And when we have 2 copies of the key gene, we will have, I'm trying to, yes, now the laser point is working. When we have 2 copies of the key gene, then we'll see a white spotted animal, as we see in the Pietran breed. And for the dominant white breeds such as Landrace and large white, we can see several combinations. We can see 2 or more copies of the key gene with a mutation in a splicing site. And also there will be duplications upstream and downstream KIT. Besides being related with coat color, CNVs are also associated with economic threats. Such an example, ZNF gene, when it has a copy number state, it can be related with test size in Q sheep. CNV in the AKAP13 gene is related with growth threats in goats. CNV within the HMGA2 gene is related with navel length in Bos indicus, and this CNV is exclusively for Bos indicus animals, is not presented, is not in the Bos taurus population. And there are also CNVs associated with reproduction and disease threats. Some studies from the CGIL have demonstrated that. The very first study of copy number was in 1976, so it's not a new area for study. And it was a study with a bacteria, the Bacillus pumilus strain. The very first study on humans was in 2002, and the first study in a livestock species was in 2009 with cattle. And how we can detect those CNVs? We can detect by using mainly 3 types of molecular techniques. We can use array comparative genomic hybridization. We can use bead sheep SNP microarrays, and also whole genome sequencing using short and long reads. The SNP arrays and the whole genome sequencing are the most common methods to identify and to detect CNVs, especially in livestock species, due to the to the possibility of doing large-scale studies. But even using high-quality information, there is a high number of false positives when CNV detection is performed. Then it's recommended to validate those CNVs. We can validate CNVs using real-time PCR or also FISH, fluorescent in situ hybridization. And those techniques will provide high accuracy and specificity, but they are time-consuming and expensive, which is a limitation for large-scale detection and large-scale studies. So an alternative, in order to not spend a lot of time and money in the lab, would be perform in silico partial validation. A CNV can be partially validated when the same region containing copy number variants is detected using two molecular techniques, such as whole genome sequencing and SRIP array data. And this will increase the CNV detection reliability. Thus, the objective of this study was to detect CNVs in the Dairy Gir cattle, to define high-confidence CNVRs using two in silico approaches, and also to functionally characterize those CNVRs. As material, we had SNP array samples and whole genome sequence samples. For the SNP array data, we first performed the quality control, and for the whole genome sequence, we performed the quality control, alignment, and several steps of post-processing. We then detected CNVs separately for each molecular technique. We then established two sets of high-confidence CNVRs, and then we performed function analysis. This is a fluxogram of the flow of our material and method, but I will explain step by step. For the whole genome sequencing samples, we accessed 38 sires from the National Dairy Gir breeding program, and the sires were selected based on their progeny number in the program. They were sequenced using Illumina platforms, and the samples, the reads were aligned to the ARS UCG 1.2 genome reference. Their coverage ranged from 10 to 25-fold with an average of 16-fold. For the SNP array samples, we accessed 566 Dairy Gir and males and females, and they were genotyped with the Illumina bovine high-density big sheep, and we only retained SNPs based on autosomal positions on the same genome reference as the whole genome sequence, so we could compare both results. For quality control purpose, we didn't include SNPs with a GenCov score less than 0.15. For both sample sets, we had 36 individuals that were both sequenced and genotyped. So same animals, but different molecular techniques. We have a molecular technique with ultra high density and another with high density information. We used two algorithms to detect CNVs from the whole genome sequencing data. The first one for whole genome sequencing data was CNVinator. CNVinator is a common algorithm used in livestock species. For the software, we performed individual identification, so each individual at a time. CNVinator uses REDAF methodology, and we established a bin size of 250 base pairs with a mean REDAF sign of 4.12. CNVinator uses the read count within each bin, within each 250 base pairs as our RG signal. And we also selected fraction of low quality mapped reads less than 0.5. We only selected CNVs with mean signal of REDAF significantly different from the average signal in the sample. We also restrained the size of the CNVs. We only selected CNVs from 1 kilobase to 5 megabases. And at the end, we overlapped CNVs into CNVRs using the criteria of overlapping of at least one base pair. The second software was DALI. DALI uses two methodologies to identify CNVs. It uses read pair methodology and split read methodology. We use some parameters to ensure that our detection could be as accurate as possible. We selected the measure of probability that a read is aligned in the wrong place as 20. And that CNV, one selected CNV that were supported by at least 4 read pairs with the same size. And at the end, we also overlapped CNVs. We merged them into CNVRs. Moving to the SNP array samples, we detect them using PAN-CNV, which is a common algorithm for SNP array data in livestock species. It uses Bayesian methodology with Hayden-Markov model. And for the SNP array, we need more than the information that is common shared, like the matrix 01. We need a file that is called final report, and that has several columns. And we need especially the information about signal intensity of the SNP, then we will call log R ratio, and also the B allele frequency in each sample for each SNP. We then adjusted the log R ratio, the signal intensity, for each SNP for genomic waves, 500 kilobase around the SNP. What are genomic waves? Genomic waves refer to signal noise related to the GC content, the guanine cytosine content. So we adjusted the signal for guanine cytosine content around the SNP. We only selected CNVs that had at least 10 SNPs, and we also selected some parameters to accurately identify CNVs. We selected this, we had the same size restriction and also merged overlapping CNVs into CNVRs. From that, after identifying CNV using two algorithms using two softwares in the whole genome sequencing, and also after identifying CNVs from the SNP array data, we can build now our high confidence CNVR sets using in silico validation. We use two approaches, and at individual level with that 36 animals that we had both information that we're gonna call CNVR any, and also with population and sets of CNVRs, CNVR top, that are referred to the CNVs that were identified in at least 5% of the population. For the first set, the individual CNV set, we then summarizing have the CNVs from CNVinator algorithm, and we had the CNV from PAN-CNV algorithm, two different molecular techniques information. We then checked if the different algorithms detected the same CNV by establish criteria of 50% of reciprocal overlap between the different algorithms. We then have now the individual CNVs that were identified by both molecular techniques. And then we merge them, establishing then the individual set, the CNVR any. For the populational set, we have to remember we detected CNVs in whole genome sequence using two algorithms. We checked if the same CNV was detected by both algorithms, and then we merged, we checked if they were, and then we had the CNVR-seq set. For the CNVRs detected in the SNP array data, and then we checked if the same CNVs were identified in whole genome sequence and SNP array data with the criteria of 50% of overlap. And then after this, we selected only those CNVRs that were present in at least 5% of the population. And then we established the CNVR pop, our population CNV. Taking a step further into trying to validate those CNVs, we performed CNVR visualization with Samplot software. For the function analysis, we merged those two sets of high confidence CNVR any and CNVR pop to see if the high confidence CNVRs were overlapping KTLs and genes. We used Gallo-R package for that. After we performed gene enrichment of the genes for gene ontology terms, metabolic pathways, and also MASH, medical subject headings. We compared the CNVs we identified, the high confidence sets, with previous studies. There is a public available database called the GVA database. We then selected CNVs from 8 studies with cattle, and then we merged overlapping CNVs into CNVRs. And then we checked if the CNVRs detected in our study was also detected by previous studies in the GVA database. As a result, one of the first results we can note is that we have a large difference of the numbers of CNVRs detected by whole genome sequence data and by SNP array data. We have a higher number here in CNV nature and DALI than in pan-CNV. And only 16 of them overlapped between the 3 algorithms. This difference happens because of the density of the genomic information. Whole genome sequence will provide much more information than SNP arrays. And as we see a large CNV in the SNP array data, actually it might be several short CNVs that are close to each other. But as the SNP array doesn't have as much density to detect small CNVs, we see a large one. Another thing is that there are regions that are not covered in the SNP array. We don't have any density of SNPs, then a CNV won't be detected in using SNP array, but will be detected using whole genome sequence. For the individual CNVR set, the CNVR any, at the end, we ended up with 45 CNVRs with a mean size of 98 kilobases. From the 45, 23 were deletions and 22 were duplications. For the CNVR pop, we detected 10 CNVRs with a mean size of 105 kilobases. From the 10, 4 were deletions, 2 were duplications, and 4 were, as we say, mixed or complex, meaning that we have deletions and duplications happening in the same region of the DNA in the population. This figure represents the CNVRs in the chromosomes, merging CNVR any and CNVR pop. We had 48 CNVRs, and here it is represented only the chromosomes with CNVRs. The chromosomes without CNVRs with no detection are not listed. We see that some chromosomes had only one detection, and for example, chromosomes S7, 9, and 12 had several CNVRs. The blue dots that we see are CNVs that only belongs to CNVR any, that means at individual level. The red one represents the CNVRs that only belongs to the CNVR POP, that is the approach for the population of CNVRs. And we see in green CNVRs that are present in both sets. For the functional analysis, we use the data merge of those sets. Again, 48 CNVRs. And from that, we identified that those CNVRs were overlapping 69 genes and 2 pseudogenes, and 156 QTLs related to production, reproduction, exterior threats, health, and milk. We found some interesting results of CNVs overlapping genes related to immunity, like the genes from the BOLA family, that is the major bovine histocompatibility complex, MHC class 2. We found also interesting MASH terms related with these genes, as T lymphocytes receptor, MHC, antibodies, antigen presentation, and also gene duplication. That means that these genes are often subjected to gene duplication. These genes are also associated with the provider load of bovine leukemia virus in cattle, and they are under selection in Brazilian LOR. So we can think that this gene may play an important role for adaptation and also tolerance to tropical disease in Brazil. We also found genes overlapping, genes related to the mammary gland, like the ROL gene. This gene encodes a protein important for initial mammary epithelial cell morphogenesis. This study was done in rats and murines. We also found CNV overlapping pleiotropic genes like GBP family. We found the 2, 4, and 6. These genes are also related to immunity. They have been pinpointed as important for intracellular parasite elimination and also implicated in the defense against Mycobacterium avium, paratuberculosis in cattle. But they have been also related with residual feed intake in Holstein cattle and stature in Chinese cattle. We also find genes related to fertility, like SAMP6 and FILIP1. CNVRs within those genes were associated with litter size in sheep. And for the CA5A gene, we found that this gene was previously related to high fertility in Holstein cattle and is highly expressed in ovaries and uterine epithelium. A big portion of the genes we found, they belong to the olfactory receptor family. And we also find GO terms as stimulus detection and olfactory receptor activity. We understand that olfaction is very important for survival and reproduction. The animals need to recognize pheromones, and olfaction also plays a huge role on progeny recognition and mate recognition. In the literature, we can see that olfactory genes are often subjected to CNV, and there's a discussion that this gene family might have evolutionary importance in cattle and other species. For the CNVR visualization, we use SemPlot. This is a figure of SemPlot software. Here we have in the x-axis the chromosomal position. In this case, I'm showing the chromosome 9. This is the CNVR 18. And the y-axis, we have the coverage, or better saying the reads depth, the number of aligned reads in the region. And here I'm exemplifying a deletion. We can see a pounder here, and then I have a decrease of reach depth of coverage in both samples. Then this analyzes for us that there's a deletion at the site. Another example of deletion we can see in the first 2 samples that we had deletions in both chromosomes, both homologous chromosomes. So we have a copy number state of 0, and in the other ones we have a copy number state of 1. And here we have now a duplication. We see a pattern, and then there is an increase in redaff, and then a decrease. You can see in those 4 samples. For the comparison of CNVRs with previous studies with the DGVA database, from the 48 high-confidence CNVRs that we found in the study, only 8 were also reported in the GVA database. This might be because there are not many studies. We only found 8 studies in this database, and those studies had few indocene cattle samples. Some had Gir, but few samples. As a conclusion, we identified 48 high-confidence CNVRs in a population of Dairy Gir cattle using in silico approaches that might be related to evolutionary process and adaptation for tropical disease. We found CNVRs encompassing genes that are responsible for phenotypical variation of important threats for the dairy industry in South America, such as lactation, fertility, and also health. This is the list of reference. Those references are not included in the article. You can watch the video and print them. And I'm happy to take any questions you might have. Thank you for your attention.
We're clapping in the room for you, Larissa. That was a great presentation. Thank you so much. It was very interesting. So now we will open up the floor to questions. So if you're online, feel free to turn on your camera and microphone. And in the room, we have some microphones if we have any questions. We have one in the room here from Bruno.
Hey, Larissa, Bruno here. Thank you for your presentation. Larissa, my question is maybe a technical question, because you have shown for us at the end of your presentation that differences in coverage to discover deletions and insertions, duplications and deletions of copy number variations. That figures. But for SNP ships, how could you tell again how it's possible to detect? Should you compare with the whole genome sequence, or only look into SNP data? Because SNP data, you can't get this information of coverage, for example. So if you have 1, 2, 3, 10 CNVs in the same animal, you get the same SNP. So at the end, you doesn't matter. You don't have the sequence per se. So how do you detect this information in SNP ships?
Okay, thank you for your question. As in whole genome sequence, we have redaff. We don't have the same information in SNP array data, but we have signal intensity. So the signal intensity, it is calculating with the platform, with the genotyping platform. So if I have a duplication, I will have an increase in signal intensity. If I have a deletion, I will have a decrease in the signal intensity. And also the B allele frequency, it will change when I have when I don't have a copy number variant, or it will increase or it will decrease. And by doing that, we can identify copy number variants. So we need the SNP, the map, the map file, and we need the SNP and its signal intensity and B allele frequency. The B allele frequency can go?
Go ahead.
The B allele frequency is a normalized measure when you have the intensity for A and B.
Okay, so we should have the final report, not only the genotype file.
Yeah, you need the final report. Usually the logger ratio and BAF, B allele frequency, usually they are the last columns. And you need that
Okay.
information.
Okay, thank you, Larissa.
Okay.
Thank you very much. I think we have a question online from Flavia.
Hello, can you hear me, Larissa?
Yes, I can.
Good. Nice to see you. And thank you for presenting to us. It was really, really nice presentation. I just like to comment something to Bruno too. That Bruno, I think I know what you are thinking about the new project that you are just starting in Brazil. So it's very important that when you kind of plan the genotyping that you ask the provider, let's say Neogen, for to send the final report with that information. Otherwise, they are going to just send the genotypes. So you have to ask, send me the complete final report. Otherwise, you get just the genotypes.
Okay, thank you, Flavio.
Yeah, no, I think maybe you are thinking to the project, right? The new project. So that's one, just a comment. But Larissa, you mentioned that you found like 35% of the genes related to olfactory system, right?
Yes, yes.
So, but you didn't mention the importance of the olfactory system for the cattle to prevent eating plants that are toxic, right?
Yes.
And that is especially important in cattle because cattle cannot really like a sheep that can, because have teeth in jaw and in the upper teeth and lower teeth, right? So they can really pick what they like to eat. With the cattle, they use the tongue and get everything that's in front of them. But if they can smell that plant, they are going to go around and prevent. So, and I think in the tropics, we have a lot of toxic plants. Could have,
Yeah.
right? So you didn't come across this type of relationship of the olfactory system and the ability to prevent to eat toxic plants. I'm
Yes.
talking about the system that they have a grazing system, right? Not like in Brazil.
Yes, we have several toxic plants that we have in the farm, in forests surrounding the farms, and yes, we have a grazing system. Yeah, great contribution, Flavio. Thank you.
Yeah, maybe pay attention to this when you read to connect the olfactory system. Maybe perhaps what we are seeing as adaptation for the tropics.
Yes.
And another thing, I just like to explore your brain. So you have these tires that were 38, I think, that were sequenced. Right?
Yes.
And they were selected based on the number of progeny. And I know that was the size with the most progeny, right?
Yes.
So, but if you like to really analyze the diversity of your CNVs, right? Do you think that was the best strategy for that particular study? Because if they are the most used size, because they have something in common, right? Probably the daughters are high producing milk. So these sires are more alike than random sires, right?
Yes, we do have father, son, grandfather in the samples. I didn't include a pedigree file or the relationship for CNV detection.
No, my point is you have, maybe you didn't have the diversity of CNV regions in your data set because you are using the most common sires, and probably they are the
Yes.
most selected ones, right?
Yes. Yes.
So maybe now you could have identified more CNVs if there were not these top sires that are heavily used in the breed. Maybe another
Yes.
strategy to sequence some other animals, like more randomly males, might produce more help the identification of CNVs more than just use this. But I know that you use this data was not planned specifically for this project, but it's something to think about when you look at the results, right?
That's
It's
true.
different from when you compare with the ones that were genotyped with the panel that are more diverse animals,
Yes,
right?
that's true. They are more diverse. Um, but I think a good point of having those sires with the most progeny number is that we might have detected CNVs that are present in a bigger portion of the population because they have
Yeah.
many progeny and grand progeny. So we might find a CNV also, the same CNV because it is in they can transmit to the progeny the same CNV.
Yes, no, and then in that point, but again, is you probably has less even views that you would have. It was a more diverse sample of animals.
Yes,
But
I
yeah,
agree with
just a
you.
comment. Very good, great work, and thanks for presenting to us. It's good to see you.
Thank you for your question. Good to see you, Professor.
Okay, and we have a question in the chat here from Saranya asking if you have any comments on the application of CNVs in the industry.
Oh, there is a paper from 2018 where they included CNVs in the model and they have a slight increase in the accuracy. But this year in ASAS, a PhD student from Florida presented work where they included CNV in the genetic evaluation, and they have, I don't remember exactly how much, but a good gain in accuracy for reproductive traits that usually have a very low heritable um, heritability value. And I don't know, maybe we will start to see more studies with CNV in genetic evaluations, but I didn't have the opportunity to find many. Another thing that I uh, think about of CNV for the industry is that they are important for in the disease part. They are really related with the disease and maybe can be the causal mutation for a disease that we didn't find yet. And also, I see that runs of homozygosity topic is growing. And when you detect runs of homozygosity with SNP data, um, you might have a confusing aspect with CNV. So CNVs might gain further focus on this.
Very interesting. Do we have any more questions online or in the room? I have a question. Flavio has a question. Did you just put your hand up again?
Yes, I put
Okay.
up again. So no, because this is a very interesting discussion about the application of the NVs. So Larissa, I have a question for you. I don't I think you never did this, but would be a very important aspect to look at because you have the sequence data, right? So the CNVs from the sequence, and you have for the same 36 animals, you have the genotype with the SNP. So do you know from the if you use the SNPs that you have in the commercial panel, how many of those the variation of the CNVs will be captured by the SNPs defect, right? Right? Because you have the CNV, but part of the CNVs, I know this for sure because I did some work for the paper that we can capture the variation of the CNVs by the SNPs. So when you feed the SNPs the model, you are capturing some CNVs, not all, but a good portion of them. Have you looked at this to see, have idea of the portion of the CNVs that you can capture with the commercial panel? Because you have a data set that could allow you to look into this.
Yes.
Because that's
No.
important for the application, right? So you
Yes.
like to include in the model just CNVs that are not already explained or captured by the SNPs.
Yeah. I didn't look into that, and I don't think there's other studies with this right now in the research group, but maybe a postdoc with that. Maybe in the future.
Yeah.
I think it would be really interesting.
And it can be done in a data set like yours that you have the animal, a group of animals genotyped with both, or not genotype with the SNP panel is sequenced. So that then you can look at this and have an idea of at least decide which CNVs are going to include in the model, which ones you don't need because it will be redundant with the fact that you have a SNPs capturing the effect. Just a comment. Yeah, idea there. Thank you.
Yeah.
Thank you. I think we have sort of a similar question in the chat here from Hamza, asking, perhaps I missed this in your presentation or published paper, given that whole genome sequence detected 325 times more CNVs than SNP panels, what would be your recommendation for breeding programs trying to balance the cost versus the detection power when choosing between these methods?
Yeah, I think with the decrease in price of whole genome sequencing, maybe we'll see more whole genome sequenced animals, even for low coverage. And a good way to detect those CNVs in a lower level of cost would be to genotype the animals and then perform in silico validation with some whole genome sequenced animals. And then you can trust more in the information that you were seeing in the SNP array. But a SNP array less than 50 K, it would be very, I'm not sure if it would be a good accurable result. I've only seen studies with at least SNP arrays with 50K.
Thank you very much. Do we have any questions online or in the room? Nope. I have a question. So I'm a little bit new to CNVs. I saw in your results that you had different density of CNVs depending on the chromosome. Is that something that you expect? Are there certain regions that are more likely to have CNVs, or is it pretty random where they come up?
So yes, we have some, I could say, CNVs hotspots, especially in immunity-related genes, because an individual might benefit from duplication in an immunity gene that can maybe result in a better immunity response or these kind of things. So yes, there's definitely genes that are more related and that are subjected to copy number variants. The core genes, however, the genes that are really essential for life, they are less subjected to copy number variants because disruption in a start codon or in a regulator or in an intron exon may cause a problem. So yes, there are definitely spots that are less subjected to copy number variants, such as those core genes, and spots that are more subjected.
Mhm. And do you see a similar pattern with the length or size of those regions? Does it vary depending on what gene it's affecting, or is it relatively random?
That I'm not sure if I can answer, but usually it's very common to find not very common, it's common to find CNVs in intergenic region where they still can affect phenotype, but they would cause less problem than actually disrupting a gene, for example. So maybe in intergenic region, they could be larger.
Very interesting. I'll open it up one more time for questions in the room or online. I think we've had a good discussion here. It's been very interesting to learn about CNVs. Okay, I think we are all set on questions. Thank you so much, Larissa.
Ella, may
Yeah.
I ask another?
Please go ahead.
Because of this discussion of where the CNVs happen. So, Larissa, do you know if CNVs will be like SNPs? They would happen less close to the centromere of the chromosomes? Or no, in this case?
I'm not sure if I can answer that because I remember when I read my master's, I was trying to look for these answers, like are CNVs more close to centromeres or to other chromosomal regions? And I'm not sure if I got into a conclusion.
Okay, yeah.
Yeah.
Probably
But
because there is no evidence then.
yeah, but we do have regions that segmental duplications happen, and I think they happen more close to the centromere or the extremities of the chromosome region. I can remember that, but not much, unfortunately. Sorry.
Okay, no, thanks.
Okay, I'll do one last call for questions if there's anything else in the audience. But if not, I think we will end it here for today. Thank you so much again, Larissa, for the presentation and the interesting discussion. This recording will be posted to YouTube and we'll send you all those details. And thank you to everyone who came and listened to the talk today. We will see everyone here again next week for another presentation. Thank you.
Thank you, everyone. Great to see you guys.
Yeah, it was so great to see you.